meters, but I think this looks good to me. And it sounds like we just had a satellite drop out because I lost chat.
Gotcha.
Yeah, I mean, if we were at 1 meter from the back of the vehicle, which is where the altitude is measured, we'd be in the dirt. So.
Yeah. No, this looks good. Good. It's consistent with previous transects.
Yep, this is perfect, guys.
Okay.
Great.
Good speed, good field of view. I think these will be easy to identify for the folks who have to do the analysis.
So is he up to speed now?
Yeah, he's at 0.35 knots.
Okay. This is better.
Perfect.
Yeah, I'm
Thank
happy
you.
to track a line. I just was being conservative.
Data, are you able to let us know when we hit 15 minutes,
Yes. Yep.
please? Thank you. And at that point, we'll check our distance over ground to make sure that we've gotten enough,
Sure.
space.
Just for
Thank
reference, we started this at 37 minutes past the hour.
Thank you.
Yeah.
For those who've been watching for a while, you get the sense that the habitat down here can be kind of patchy. We have a lot of sediment coming off the shelf and flowing down through the canyons. And when we do find hard bottom, there tends to be a lot of things colonizing it, like the big bubblegum corals that we've seen this morning, and the peristanella corals, and some of the other interesting species here. I invite you all to pay a particular attention to the sponges, and hopefully Abby will continue to provide her expertise during this dive. She's our sponge expert, and many of the species have very few samples that have been taken, and some of them have yet to be described. So it's pretty exciting to be exploring these habitats where there's the potential to encounter things that we don't have names for yet. Kind of a rare opportunity in the life of scientists. I also wanted to mention, while we have a little time, a few things that you should all know about a sponge expert in our region who recently passed away. Dr. Henry Ricewig has been a legend in taxonomy of sponges, and really lost an encyclopedia of knowledge in that man when he passed away earlier this year. Hopefully in future dives, when Abby's narrating, she can share a little bit about her experiences working with Dr. Ricewig over the years. How are we doing for speed, ROV?
I think we're
Yeah.
doing a pretty steady speed now.
Yeah, we've been super
Perfect.
consistent.
Thank you. Could we pan up just slightly, please? I want to make sure we're not going to run into anything as we're ascending. Looks like you have good altitude.
Yeah, this is that last part of the little outcrop.
Okay, great. Yeah, because sometimes we come up across some big bubblegum corals and things.
Did you want to kind of veer off to the rocks, or are we still holding a steady line?
Let's focus on the wall to the left.
Okay.
So
I'm gonna
maybe if we can pan
change
a
it to
little
1.5.
bit as we're moving along it, we're kind of out of the visible range of.
I'm gonna bring our line up a little north, so we're a little more over slope, or upslope,
Okay.
if that helps.
Yeah.
Great, thanks.
Yeah. Watch your. Yeah. What's your altitude? Yeah.
Now I have a request. Give a second.
For nav?
Yep. Yeah.
Yeah.
Can you on that initial that aborted transect 5? Can you just add aborted?
Yep.
To that either the description or the name.
Okay.
Good catch.
The other one I called restart.
That's fine.
Another skate tucked in there, it looks like. Raja. I believe
Bridge
is the
nav.
genus. Ooh, what's that? ROV, what's our altitude off that wall, please?
A lot of stuff raining down from above here.
Yes, marine snow is thick. Bringing lots of nutrients from the surface down to these deep sea ecosystems.
Yeah, we're pretty close there. Yeah, I will 10 more up. All right.
We're
Yeah,
about
I can bring
10
us.
minutes into our transect right now. Seems like we're doing well with the speed, which is great. Staying ahead of the dust cloud. There's a nice grenadier fish on the right. Haven't seen nearly as many hagfish as I was anticipating.
Jacob, is your altitude staying pretty consistent?
We saw some beautiful halibut on the methane seep dive yesterday, the beginning of that. Lovely. You may have seen that in the replay. I'm loving that 12-hour slider bar. Great feature on the website. Nav, when you get a minute, can you let me know how much distance we have covered since the start of the transect, please? Just for a check-in.
Yeah, we've been, it's about 110 meters from the beginning of this real transect.
Thank you. So maybe another 40 or 50 meters.
Sounds good.
We should be at our minimum time and distance. It's like we're getting into a slightly more densely colonized area with some different things that we haven't seen so much in the muddy habitats. It's a little bit patchy though, or very patchy, I should say.
Definitely looks like there's enough food for life.
And something, a little highlight from the past and coming up is in Monterey Bay National Marine Sanctuary, they identified a whale fall last year. And the bonanza that
Bridge nav.
the contribution of a whale carcass makes to deep sea ecosystems is quite impressive. So I encourage you to look back at that. There's a photo mosaic of the whale fall, and I believe they'll be visiting that site again in a couple of weeks when
So
they
we're gonna head
bring
north.
in our sanctuaries to the south.
Yeah, I've been slowly incrementing us up north just because that's the next target.
Okay.
Yeah, so now it's due north.
All right, Jenny, that's about time for 15 minutes. What do you want to do?
Let's go about another 20 meters and then call it, please. Then we will switch to exploratory mode and find a line up that looks good.
Yeah, we're about 150 meters, like crow's flight from the start of the transect.
Great, thanks. We'll just add another couple tens of meters for
Roger.
good measure.
Yeah, very close to the next target.
Awesome, thank you.
Clam patch there. A little bit. Clams.
Clams?
Clams.
It
Eric?
looks like it.
A couple of clams.
Mama and babies.
Are we about 170 meters now?
Give me 1 second.
Thank you.
Yeah, we're like 165. And we've covered more than that because that's just the straight line. But yep.
All right, thank you. Let's go 1 more minute, and then we'll stop with our transect.
Great.
Thank you. So ROV pilots, a question for you. How's the current down there?
How's the current? Well, it's varying
Yeah,
here. I mean, you can see it's been shifting. You know.
I was noting this morning how the current seemed to be a factor in where we were finding organisms. It seems like where there's food, right?
Yeah. Generally, the corals tend to be on the edges of the ridges where the current's whipping by more.
All right, let's call the transect, please.
Great.
Throw a marker with the label. Now, I'd like to go over, if we can avoid the cloud, and do a close-up on one of those primnoid corals in
Bridge.
front of us. That's possible. Maybe
The one
not
in
because we're
front of
moving
us,
too
because I don't
fast.
want to back up, because if we back up,
That's
we'll go
okay.
into our
Don't back
cloud.
up. Yep, don't back up. We'll find another one.
Yeah, go ahead.
But now that we're in exploratory mode, let's do
Yeah,
some
I just need
some
1
photo
second, and
shots and
if you can stop tracking the line right now, that would be helpful. Great, thank you.
Sorry, you wanted to see a coral or a crinoid? What were we looking at?
oh, that's okay. It was a primnoid coral that we were looking at. That we crossed over. Actually, could you zoom on that yellowish coral down on the right, please? It's just below the lasers. We're getting some noise on the SPL.
I might be breathing heavy.
It looked like an advertisement.
Really?
Oh, that wasn't me.
I don't hear that on the ship.
Sorry.
So Bob, you see that for the next target, it's gonna just not be upslope.
Sorry, what?
Yeah, I just want to show you the next target is just gonna be mostly along the contour,
Okay.
just so you know.
I was also intrigued this morning watching how at many of the coral locations there were also sponges and lots of other organisms tucked up in there. And I was wondering about how the crabs and shrimp were using the vase sponges and barrel sponges kind of as a little condo where they could hang out of the current, maybe not have to hang on quite so much. Did you guys notice that? Were you watching how seemed to be concentrations of life all together?
I didn't notice much. I really just started tuning in this morning when we were at the clam bed. So I haven't seen too much of the big corals and sponges that everyone was talking about earlier. But that would make sense, right? That organisms that might feed in the water column, suspension feeding, might want to get up and into the flow, standing on
Nav,
top
if
of a
you want to
coral or
put in
sponge,
a
and they could
ship
use that
move
as habitat as well.
20 meters at bearing 010. Okay, great. Thank you.
Yeah, it makes a lot of sense. It's sometimes hard to predict what currents are going to be like on the bottom, and there seem to be some areas that have high currents near areas that are relatively calm. I think that may be part of what's structuring life down here and the patchiness.
Yeah, there's a lot of dynamics going on. You have sediment, flowing down walls of canyons over time. You have variable currents, so it absolutely makes sense that that would be a feature of these habitats that organisms might want to take advantage of.
Yeah, we're seeing some grenadier fish off in the distance, teasing us a little bit. And then about lower center, we're seeing some thorny heads. They are well adapted to low oxygen environments that we find in deep sea areas. What's our approximate depth, please?
We're at 1190
You want
meters,
to zoom in on that, Dave?
1200
Yes,
meters.
please. Thank you. Yeah, we're really deep down here.
Oh, wow.
Off the coast, there is an oxygen minimum zone that is at approximately 1,000 meters, but I suspect that this environment does experience low oxygen conditions quite frequently. It's a beautiful zoom. It's,
It's an anemone on the coral? Is that?
Yeah, there's an anemone on the coral, and I wasn't sure in the last one we were zooming in on, but this looks to be Swiftia, which is an octocoral, Flexaurid octocoral.
Cool. Some rockfishy looking thing.
yeah, that's a thorny head.
Oh, cool.
Beautiful. Thank you. This is
Okay,
a
zoom out,
fairly
Dave.
common species in our region, Swiftia. Let's go wide and continue on. For those of you just joining us, we are working in an area called Biogenic 2 Essential Fish Habitat area. Essential Fish Habitat has been designated for protections from bottom contact fishing by the Pacific Fishery Management Council. So I expect that they will be very excited to see the results of our efforts this morning. We started this dive at midnight and now at 9 o'clock have 9 hours under our belt. And a whole new crop of folks who are waking up in the US and in Alaska and the islands. Can we get a quick check-in on where folks are joining us from? I'm not sure if we're able to do that without our science communication fellows on board. Awesome. Well, we will wait then until we have someone from that team who can weigh in. We often have folks calling in from virtually every continent. I'm not sure there have been Antarctic viewers before, but every other continent has been well represented.
Do you think that's the same species of sole, just a younger version of it? The coloration looks a little different.
Wanna zoom in, Dave?
How do you tease out those two? Just visually. Bridge now.
Okay, zoom out.
FYI for folks local, we're satellite dropped out again.
I was just able to join back again, so good news.
We got over here. Looks like a whole patch of clams.
Oh yeah.
Wanna zoom in on that, Dave?
So they must like settle from the larval stage into these spots, and then these are the only ones that are successful, I think.
Yeah, it's
Those
nice live
those are
clam
happy
bed.
clams.
Definitely. Oh, you can see their little like
Yeah,
tubes,
Siphons.
they look
siphons,
very
yeah.
healthy.
I'm just here for the
Hungry.
vocabulary, folks.
We'll have a spelling bee later.
All right, zoom back out.
Yeah, let's zoom out and keep it moving. I'm not hearing Andrew jump in saying, I need a sample. So let's keep going. That's actually one of the really fun things about this cruise is that because we wanted to work in the same area, Andrew and I were able to team up with Ocean Exploration Trust and combine our days at sea to work together. So it's been a nice collaboration. And I'm hoping that some of the additional sampling that we're able to do opportunistically will
Can
help
you
his
steal, bub?
project as
Yeah,
well.
do whatever you want. Go for it.
Awesome. Well, welcome to everyone. Welcome to Olympic Coast. This is an area off the west coast
Can
of
we do a quick
Washington
zoom there,
State.
Dave?
We are at about 1,200 meters of water depth in an area that hasn't been explored by remotely operated underwater vehicles to our knowledge. Looks like there's a little crab tucked up under there. Maybe a, oh, look at that. Beautiful.
Now, do those fragmented corals have a chance of living?
Reattaching.
Yeah, no, I mean, if it falls over, it's
Dunzo.
pretty much, that's it.
Oh well, it looks like I had a good run.
Yeah, especially in this heavily sedimented environment, that'll be
Oh right, quickly.
Hey, zoom out, Dave.
that'll be smothered pretty quickly.
Because I know with shallow water corridors, sometimes the fragmentation is part of the reproduction, like how they disperse and stuff.
All right, let's looks like there are a few fairly large colonies down there. So let's pan to the port, please, and check out
Okay.
this wall. Maybe we can go a little bit more slowly and do some Photoshop. Zoom. Lovely. I know that some of the bubblegum coral can actually be aged from the base. So getting good shots of the base with the lasers on will be helpful to folks who study deep coral species. Looks like we have a nice little cluster here of organisms.
Bridge nav, you can stop this ship move right
Jenny,
now.
I'm just talking to Meredith in the
Yes,
chat, and
please.
she did indicate that if we have a chance, she did want one of those Swiftia, or some of it.
Okay, great. It looks like down below there was something unusual, sort of a yellow looking sponge. I don't think we can get back down there though, can we?
I don't know how clear it's gonna be. No, no.